Kia ora koutou, ko Dwayne Peltzer toka ingoa, and welcome this morning to a Biosecurity Bonanza talk. Just introduction, I'm a portfolio leader for our Managing Invasive Species group. That's Manaaki Whenua Group within the new BSI. So today we're going to hear from Bevan Weir. He's a research priority area leader. He's going to talk to us today about nationally significant collections and databases, and really their role in supporting biosecurity. His background's really, can ask him all sorts of questions later about fungi, microbes, molecular. It's a little bit of a grab bag of topics. It sort of shows, I guess, Bevan's breadth of knowledge and experience as well. So I'll just give you a few housekeeping tips before we get in and hear more from Bevan. So I guess first up for these webinars, they're really a chance to ask experts in an area particular questions. So please use the Q&A function and we'll get to your questions at the after the presentation. If we can't get to your question, there'll be a chance to get written responses after today's session. It will be recorded. So if you've missed the session or want to share it, you can catch it up later. Yes, so there will be a transcription and we can share that post webinar. I'll hand it over to Bevan now, and thanks so much.
Cheers, thanks, Dwayne. Yeah, so this morning I'll be talking about these nationally significant collections and databases that we have and how they can support biosecurity. So the first question probably is, what is a nationally significant collection and database, and how did these arise and where do they come from? So currently there's 26 of these throughout the country, and collectively they're funded by MB to about $22 million per year. In the CRIs, we use this mechanism, SIF infrastructure of how they're funded. The collections themselves can date back over 100 years, but this current system was set up in 1993 after the breakup of the DSIR into the different CRIs. These next two bullet points I've just pulled directly from the MB website. So it's their words, and it's the databases and collections are critical for New Zealand science to deliver public benefit, and that these benefits accrue to many varied users and third-party beneficiaries. So really this is just saying that these collections and databases, MB is specifically funding them because they have a great benefit to the entire of New Zealand Inc., rather than just the institutions that are hosting them. So in the Biosecurity Science Institute, there are 11 of these nationally significant collections, and today I'm just going to be talking about 4 of them managed by Manaaki Whenua Group, and the ones that are probably the most used for biosecurity. So first up is CHR, the Allen Herbarium, and I'll be using these acronyms CHR and NZAC and so on. They have an interesting history and they're sort of locked in place. So CHR is the sort of herbarium code, and it relates to Christchurch. So the Allen Herbarium is essentially dried plants. So it's dried plants, and they're normally like this, mounted on sheets of paper. There's a bunch of other samples as well, ethanol samples and DNA and so on. But this is the core of the collection, and there's about 800,000 of these, of which about 370,000 are databased. And databasing in this context means taking a photo of it and transcribing all of that information you can see on there into the label, from the label into a database. And you know, that label information could be handwritten or typed depending on the age of the specimen. And of course this is based in our Lincoln site and established 1928, so we're quite looking forward to celebrating the centenary of this collection in a few years. So an example sort of case study of how the collection could be used and has been used, and this is just one of many, is fireweed. So this is a very aggressive weed which is toxic to livestock, particularly horses and cattle, and can lead to liver damage and death. And this weed's native to southern Africa and Madagascar. But in May of 2017, some specimens were sent to the vivarium from Northland. And this is a kind of common thing that happens. Someone, a dock ranger or a member of the public or a regional council person sees something unusual and they'll send it to one of the collections for storage and identification. And in this case, we conducted some DNA sequencing of it and found this was definitely a new thing, so a new incursion into New Zealand. And of course, then we preserved this specimen voucher. So that did a few things. And so obviously having that genetic sequencing locked down exactly what that species was, and we're able to record it as a new weed to New Zealand and monitor its distribution as it unfortunately spread. And of course, having this confirmed identification allows regulatory decisions by MPI and councils about what they're then going to do with this. The interesting thing, I think, is that these collections is not just static, so it's not just popped in a drawer and then forgotten about, that these continue to be used. And so these specimens were used by Australian researchers later, taking these DNA and the morphology and looking at the entire Senecio genus and looking at the delimitation of these species boundaries. The next collection that I want to look at is PDD, or the New Zealand Fungarium. And in this case, PDD comes from the old Plant Diseases Division of DSIR. This is an interesting collection. There's not many like it. It's a collection of dried mushrooms. And so this is a mushroom that I collected and took a photo of a few years ago, a very interesting Cortinarius species that doesn't open up. So it's always closed and the spores are sort of enclosed in that sequestrate form there. So we've got about 115,000 of these dried fungal samples, but also quite a substantial collection of plant diseases as well. So you can imagine a plant disease like a leaf spot or a canker, all of those are also stored in the collection. This one is based up in Auckland at our Tamaki office. And yeah, it's been around for well over 100 years. So the interesting example that I want to think about here is we're normally thinking about border biosecurity and what's coming in. But of course, we're exporting a lot of stuff as well, and other countries have their own border biosecurity requirements. So in 1991, a guy, William Hartwell, was looking at avocado in New Zealand and noticed a scab and considered this was a new record, and so published a report on this, which was commonly done. And this was a fungus, Sphaeciloma persei, a pathogen of avocado fruit. Like a really good scientist, he published a paper and then popped that specimen into PDD and the living culture into ICMP that we'll talk about in a few minutes. And obviously at the time, the 90s, the identification was based on morphology. Now, 10 years later, there was a bit of a problem in that New Zealand avocado exports into Australia were stopped. They were intercepted by the border agency and held because the avocados had scab on them. And then they looked and saw that in the literature that New Zealand had this record of Sphaeciloma persei pathogen. So they blocked it at the border. Now, we and Kerry Everett from Plant and Food Group did a lot of this work, went back, and this is the advantage of having collections, we went back to that exact sample that William Hartwell had identified and had a look at it. And we also looked at the culture as well. Obviously, you know, a few years on and we could do DNA sequencing now, and the culture turned out to be a completely different species. Well, that's a good point, but you know, that could have been a contamination or he just deposited the wrong thing. And so then we looked at the actual avocado skins themselves. And the advantage of PDD is that we have New Zealand material, but also reference material from common known diseases from all around the world. And so we're able to compare the real avocado scab pathogen and the one that William Hartwell identified side by side, and putting this together with the DNA evidence, we and Kerry certainly concluded that this was a misidentification, that this avocado pathogen wasn't present in New Zealand at all, and that actually these symptoms were most likely due to wind rub, abrasion of the skin. And this allowed avocados to be exported back into Australia, opening up that whole export pathway again, which was a fantastic result for the avocado growers. The next collection, I sort of alluded to it there in that previous example, is the ICMP, and in this case the acronym actually stands for something, the International Collection of Microorganisms from Plants, although I just call it the ICMP Culture Collection, and so this is the one that I'm the curator of. So this collection is quite different from all of the others in that it's a living collection, and so we keep these everything in here, which is fungi, yeasts, bacteria, and chromista. So these are the plant pathogenic chromista like phytophthora. We keep them alive. And this can be quite a challenge, of course, because how do you keep something alive for 100 years, like all the other collections have been around? We've tried a few different methods over the years, but since the year 2000, we've been using liquid nitrogen. Which is obviously very, very cold and can keep them in a state of cryopreservation. So up at the top right there is an agar plate of, that's actually E. coli that I isolated from a duck pond about 20 years ago. And down the bottom there is the liquid nitrogen tanks. So the ones off into the right-hand side are the big tanks of pure liquid nitrogen, and then they get fed into those other larger tanks at the back where all the samples are kept. And we use a cryoprotectant to make sure that they can, when they freeze, that the ice crystals don't pop the cells. So this collection is quite varied, and so we've got a lot of plant pathogens, of course. The cultivated mushrooms that are able to be stored in there, and various environmental isolates as well. This collection is one of the top 3 in the world for bacterial plant pathogens. So almost every single bacterial plant disease that has ever been described, we've got available in this collection for use for researchers in New Zealand and all around the world. It's a bit smaller than the other collections with the sort of intense work that goes into storing these. So we've got about 25,000 cultures in our Auckland office, and it's a little bit younger, established in the 50s. So this collection can be used for many, many different things, but one example, and looking at now sort of biosecurity of native ecosystems, is this Kauri dieback, which many of you will be familiar with. So in 2006, Peter Madison noticed that some kauri trees in the Waitakere Ranges were dying, and they were progressing with these symptoms of bleeding trunks. So cutting a sort of a very long story short, we investigated this and looked at what we could isolate from the roots and from these bleeding lesions, and found a Phytophthora species that didn't really match anything that we'd seen before. So the sequence looked like one species, and the morphology looked like another species. And of course, we went back and tracked historic records of diseased kauri, and found these records from the 1970s on Great Barrier Island, and had a culture from that time, and it matched what was seen in the 2006 Waitakere samples. So we did a lot of taxonomic work on this, and determined that it was a new species, which I called Phytophthora acalycida. The good thing that we had with the collection, of course, those historic samples going back to find out what it was at that original point in time. And all of these cultures is available for use in the future. So the sort of underpinning taxonomic work and the collection, it's been fantastic for me seeing uses I would never have thought of. So some of these cultures being used to train detector dogs that can sniff out this disease. So yeah, having these collections and taxonomic work sort of underpinning that core biosecurity, which can then be used for other uses as well. Another example, this time switching to a bacterium, was the PSA disease of kiwi fruit. So in November 2010, growers in Te reported these leaf spots, and the image on the right-hand side there is this is actually a kiwi fruit leaf taken exactly at that time. But this type of pattern is really classical for a bacterial leaf pathogen. You can see it has these angular margins and doesn't cross over the leaf venation. So MPI, or MAF it was known, of course, at the time, identified this bacterium as PSA. And they used our collection to do this. So we had the type culture. So when Takikawa from Japan described this as a new disease in the 1990s, he deposited his bacterium, the type culture, into our collection. And this happens quite a lot. Just even last week, I had someone from Jamaica sending in some new species to be stored in the collection. But the key question at the time was this new to New Zealand, or was it something to do with changing environmental conditions? It might seem like an odd question now, but at the time we really didn't know, and that growers had reported leaf spots in the past, and this disease hadn't progressed to the canker stage. And it was a very wet spring that year. So there was some thought initially that, well, has this been around for a while and it's just, some environmental conditions that are kicking this off. So what we did is we went back into ICMP and looked at all of the bacterial isolates that were found from kiwi fruit and screened them all over the past 40 years and found no matches to PSA. So we concluded that it was a new incursion. Now we did a bunch of other work on looking at the populations of these bacterium and found low and high pathogenicity. The final collection that I want to talk about today is NZAC, or the New Zealand Arthropod collection. So this is a collection of arthropods, or invertebrates. So not just insects in the strict sense, of course. There's spiders and mites and nematodes and all sorts of other things in there. Most of the collection, in terms of space, is these about 1 million, maybe 1.6 million pinned specimens. So just like this insect here, in a drawer, pinned out, dried with a, keeping it preserved and intact, dried. We also have another substantial collection in ethanol as well. Again, this is currently based in Auckland. It started in the 1920s and then in Nelson, and then moved up to Auckland in the 1970s. Now, with this collection, we've got all those same stories that I've just gone through with the other ones, but what I'll talk about is something a little bit different. So once you've got these collections and you've databased and you've taken photos of many of these specimens, you can start to do interesting things. And one of them that you could use is using this sort of deep learning for automated identification. This is kind of either in the diagnostic sense or part of surveillance activities. And so the first example there is looking at BMSB, or brown marmorated stink bug, a pest that we certainly don't want in the country. And so in this study, we looked at using images of these and compared to other stink bugs and how accurate a model trained on these would be. And we found about 93% of the images were correctly identified. Now you can imagine that in surveillance scenario, this would really speed up getting some identifications. I'm sure you've all used iNaturalist or other apps that can identify insects and birds and fungi and trees. But this is really taking a subset of images and very precise. And so for the second example here, we're looking at pest flies, but just looking at the diagnostic features of their wings and got a level of 98% accuracy over 34 different species of pest flies. Now, I mentioned that there was also these ethanol samples at NZAC, and so this third image there is one of these from the 90s. They're poured out onto a plate and capturing that entire image, and then both counting what was there. And in this example, the count was about 95% accurate, and then also identifying everything that was there all in one go. And that was about 92% accurate for this example. And this was recently published by Aaron Harmer. So it's an interesting use of some of the data that we can get out of these collections, as well as the specimens themselves. So then the question is, well, how do we access this data? And of course, you can contact me or one of the collection curators, but we have two key websites delivering this data. And so one we call the SCD, which is Systematics Collections Data. This is just data on each individual specimen. So each specimen has its own web page, all sorts of information like collector, location, images, DNA sequences, all that type of thing. And we've got about 720,000 of those currently databased over all of those collections. The other website is Biota NZ, and so this is a bit different. And so this is focused on names, literature, and taxon concepts. So a concept is what somebody thought a species meant at some point in time, which can change. And the most common example of that is during the early European colonization of New Zealand, people put European names on mushrooms that were actually endemic New Zealand things. We've got about 160,000 names in there, and it's constantly updated with the taxonomy and synonymy. And really importantly, the biostatus. So is this found in New Zealand or not, or is it endemic, exotic, and so on? So in summary, I've presented just a small selection of case studies from these collections, and hopefully presented that they are really a powerful resource for biosecurity, and that they're not just passive records like stuck in a museum drawer or something. They are real active research tools used for biosecurity research constantly. And the advantage of them is that they're well curated, so we really put a lot of effort into identifying these and keeping them up to date, and that the data coming out of it is also really high quality as well. And backing all of these collections, expert taxonomists in their field and biologists who understand these organisms. One of the fun things and useful things that I think we can do is this ability to time travel. We can go back into the past because these collections are historic and see what something was like at some point in time and then re-identify that, which of course we can't do that with paper records like that paper that William Hartle published on the avocado. If he never deposited his specimens, we would have been stuck with that paper record. And the same is the case today with iNaturalist observations. Having specimens, we can actually verify them. So in terms of impact, we've got these collections support diagnostic surveillance. They can inform biosecurity policy about what's present or absent in New Zealand, and ultimately play an important role in the biosecurity ecosystem of protecting the productive sector and the natural system from weeds, pests, and diseases. So thank you for listening, and thanks in particular to Inez Schumberger, Marge Padamsi, and Darren Ward from the other collections for providing me some information, and happy to take questions.
Fabulous, Bevan. Thank you very much. And we have a few questions and time to get into them. So one we'll start with is, kia ora, Bevan, do you just collect rDNA sequence data for each species, or is it much broader than that?
Yeah, it's a good point. So rDNA, so this is more commonly probably known as barcoding. So do we just do barcode genes or more? It's variable. So for the culture collection, because bacteria and fungi are quite hard to identify, we do, as an initial step, just do that single barcode to get at least to a genus level ID. For the plant collection, there's a couple of different barcode genes. So yes, so for the culture collection and for the fungarium, we do routinely barcode. For the other collections, not always. More deeper than that is on a case-by-case basis, sort of as a research project rather than a collection activity.
Fabulous. We've got a slightly different take. So this is a question about the biostatus, Bevan. So it's, can you estimate what proportion of the work in the collections is dedicated to biosecurity versus indigenous biodiversity? And that's, yeah, I'll leave it with you to interpret. That's a quite a difficult one, but maybe around biostatus, and I guess what effort is put into the documenting indigenous biodiversity versus discovering introduced organisms, perhaps.
Yes, so we do split collection activities from research activities. So some of that work would be as research projects, and that would be the normal way of how we decide when research projects are funded. For biostatus, it's something that we're very happy to update on a needs basis. So if you think something might be incorrect, or if you notice a new paper coming out, we're happy to investigate and look at it. And then we run specific projects. So I've got one going now looking at certain discrepancies in the database. So yeah, it's I guess there's a huge number. Like I say, there's 160,000 names. And so you really have to batch it up into certain chunks of time working on aspects of it. You just can't keep everything all up to date all at once.
Fabulous. Now, there's a couple in the chat, so we're just going to dive into that. There's one around the automated identification, Bevan. So, kia ora, Bevan, do you foresee automated species identification or deep learning tech eventually replacing genetic methods for identifying non-cryptic species or unwanted organisms?
All right. I see the qualification there, non-cryptic species. Yeah. Look, I've been at this game 20 plus years, and techniques always change. And you know, as scientists, we adopt and integrate all of the latest techniques, but that doesn't mean we forget the old stuff as well. So when I'm describing a new fungal species, absolutely, I'm using genomic sequences, DNA sequences, but also looking at that morphology as well. So I don't necessarily think it's in terms of completely replacing, but it's just another tool that gets integrated into the toolbox.
Great. Got a question here, Bevan, about restoration and native plants. So could you use the databases and collections, I suppose, to guide what plant was growing or associated with other plants for guiding restoration, I suppose?
Yes, you can. We also have another database that I didn't talk about, NIVS, the National Vegetation Survey, which is a much more sort of comprehensive at that data level about what plants are growing where and what are associated with the others in different habitats. So yes, I think it's a mix of different databases and potentially iNaturalist in there as well.
Hey, that's excellent. So we're just kind of at the wrap up now. Bevan, is there any sort of parting thoughts, things that maybe come to mind you want to leave people with?
Really, you know, these are collections and databases for everyone, and we're keen for them to be used. And so, yeah, if you think that some of these examples that I've gone through today are useful to you, very happy to get in touch and discuss more.
Fabulous. Hey, so just a reminder, if you do have other questions for Bevan, put them in the Q&A as idea or the chat, and we'll follow up as needed. But just want to thank everybody for making it this morning to the Bonanza. Ngāmihi to Bevan for such an awesome presentation. Cheers, Bevan, and Adrian for keeping things running smoothly in the background. Thank you very much. Also, there'll be a copy of the webinar and transcript available online in due course. So on behalf of the BSA team, thank you to everybody for attending. See you again. Ka kite.
Ka kite.